In the neurobiology research system, breakthroughs in the field of taste perception are considered especially awesome. In this video, the members of the research team are part of an elite squad called the Special Taste Receptor Team. This is their story.
Hey, how are you? You look puzzled.
I've just been thinking a lot about taste lately.
How so?
I was wondering if the activation of specific taste receptor cells provides specific taste perceptions, or if it's just the receptor itself that determines our perception of taste.
That's a great question.
Let's go try to figure it out!
Hey, wait for me.
Scientists have long known that there are 5 different modalities of taste: bitter, sweet, sour, savory, and salty. However, scientists didn't know why we perceive these different tastes. Is it an intrinsic property of different taste receptors, or are there different categories of taste cells that influence how we perceive the 5 modalities? This study looks at sweet and bitter cells.
This is phenyl beta diglucopyranoside, or PDG as we like to call it. This substance tastes bitter to humans, but does not elicit a response in mice because they lack the receptor that binds PDG.
This is the human PDG receptor. It's called HT2R16. In this experiment, we are looking to see if this receptor is responsible for the bitter taste response, or if the receptor cell itself is responsible.
Allow me to introduce you to some of the mice that are going to be helping us out today. This is mouse 1. This is mouse 2. Now I'm going to be genetically modifying them with the HT2R16 receptor. This receptor is normally only found in humans.
The scientists created 2 types of transgenic mice using the HT2R16 receptor.
The transcription of this receptor is going to be driven by the promoter that usually drives T2R receptor expression. This receptor is normally found in cells that respond to bitter taste.
The transcription of this receptor is going to be driven by the promoter that usually drives T1R2 receptor expression. This receptor is normally found in cells that respond to sweet taste.
And so the experiment begins. Let's introduce our players. Here we see the bitter cell transgenic mouse. Her receptor is driven by cells that respond to bitter taste. When given a choice between water and PDG water, this mouse shows a high avoidance of the PDG water and much prefers the regular water. Here we see the sweet cell transgenic mouse. Her receptor is driven by cells that respond to sweet taste. When given a choice between water and PDG water, this mouse highly prefers the PDG water in comparison to the regular water. Here we see our control mouse. She has no expression of the PDG receptor since she has not been subjected to transgenics. She seems to show no preference to the water or the PDG water since both taste the same to her. The scientists diagrammed the results of this experiment on a graph with the PDG concentration on the x-axis and the percent total consumption on the y-axis. The results showed that the control mice, which lacked the PDG receptor altogether, showed no preference for either the PDG or the water. This is represented by the black line on the graph. The mice that express the PDG receptors driven by promoters found in the bitter taste cells consistently avoided water containing PDG. This is what you would expect to find, since PDG in humans elicits the same response. This is represented by the red line on the graph. However, when mice expressing PDG receptors driven by promoters found in sweet taste cells were given the same choice, their preferences differed. The sweet cell mice were attracted to the PDG to the same extent that the bitter cell mice were repelled by it. This is represented by the green line on the graph. These results clearly show that the perception of different tastes depend on which category of taste cell, as in sweet or bitter, is activated, and does not actually depend on the molecule that elicited the response. The big take-home message here is that taste perception is driven by certain cells designated for each taste modality. Let's take a bitter taste, for instance. Rather than this receptor being present in every taste cell, it would only be present in cells that register as bitter taste. Thanks for watching, and good luck on the exam!